Hello students, welcome to EPG Pathshala. I am Dr. Parvesh Sharbar from Biochemistry discipline, Indira Gandhi National Open University. Today we are going to talk on the module Biotherapy of Fungal Diseases under the paper Molecular Therapeutics. As you know, fungal infections, they can affect anyone, but their threat is increasing day by day. Now, what does the term biotherapy indicates? The term biotherapy refers to the treatment to restore the ability of the immune system to fight the infection of disease. This unit on biotherapy of fungal diseases will give you an overview of therapeutic strategies that are available to fight against major fungal diseases or mycoses that threaten human health. Especially this unit enlists 5 classes of compounds. It gives an overview of these compounds that act as antifungal agents. Students will gain useful information on their broad structure, mechanism of action, and spectrum of activity. The students are expected to identify properties of these antifungal agents, which we are going to discuss. They have to determine the usefulness of the drug discovery program, especially why we are stressing this on usefulness of drug discovery program, because these drug discovery programs in reputed labs have led to the discovery and development of these antifungal agents. We will also discuss, and you are also expected to discuss, the mode of activity of these antifungal agents. Students will also be expected to consult references and literature related to newer antifungal approaches. Biotherapy of fungal diseases. The term biotherapy refers to the treatment to restore the ability of immune system to fight the infection of the disease. Consider the fungal infections. The number is very small. Why their number is small? The number is small because in normal healthy individuals, the intact immune system offers effective resistance to fungal pathogens. So, fungal diseases are often a function of immunological deficits found in the host organism. The high mortality rate is especially seen in case of fungal infections in Candida and Aspergillus. This mortality rate, especially in case of invasive fungal infections, is very alarming. Despite the new technical advances in the field of diagnostic and therapeutics, this figure is increasing day by day. In the developing world, as per the data available, there are about 1 million cases of cryptococcal disease per year, and it results in about 6,75,000 deaths. Another area of concern is allergic fungal syndrome are also being increasingly recognized. So, there is urgent need to improve the therapeutic options in fungal diseases. Going back to the history of the fungal infections treatment, the treatment of fungal infections with potassium iodide began in 1907. This is the historical background. But until now, a lot of antifungal agents have been discovered. A large family of these compounds are existing, but there are several problems in usage of antifungal therapy, which limits its usage. So what are the problems? Number one, fungi, as you know, are eukaryotic cells, and mammalian cells are also eukaryotic. So the antifungal agent will not be able to discriminate between the fungal and the host cell, and it will lead to disruption of both of them. Second limitation, toxic side effects may occur during treatment with antifungal drugs. Antifungal agents decrease solubility, and their poor absorption in gastrointestinal tract is also one of the key area of concern as far as their usage is concerned. Next issue is their lower penetration and distribution, especially in the affected areas, and there are limited number of molecular targets. So at present there is a very limited range of antifungal products that are available in the market for therapeutic purposes. The antifungal agents which are used for therapeutic purposes are generally they are classified, though their number is much more and new ones are coming up in the development. Generally they are classified into 5 classes, right, for the treatment of fungal infections. Now each of these 5 classes of compounds have their own advantages and disadvantages. Antifungal agents, they can be naturally or chemically synthesized. They can inhibit the growth, the term is used fungistatic, or they can kill the cells, the term is fungicidal. So the 5 classes of antifungal agents which are discussed in this module are Polyene antifungal agents, Azoles, flucytosine or fluorocytosine, allylamine and thiocarbamates, and Echinocandins. Polyene antifungal agents. The first Polyene antifungal agent, called nystine, was discovered in 1950 by Rachel Brown and Elizabeth Hazen. This discovery was followed by the discovery of other antifungal agents such as recidin, remocidin, endomycin, ascocin, trichomycin, and antimycocin. Then amphotericin A and amphotericin B antifungal agents were isolated from Streptomyces nodosum, and this discovery was made in the year 1955. However, out of these two, amphotericin A and amphotericin B was further developed because it was having greater potency than amphotericin A. About 20 antifungal agents of this class are today known to exist. But there are several issues associated with their development. Some of these issues include stability, solubility, toxicity, and bioavailability. So there are very few polyenes that are available for the treatment of fungal infections. Especially in developed countries like USA and UK, authorities have given their approval only to amphotericin B and nystatin for therapeutic purposes. Nystatin is helpful for superficial mycosis, and amphotericin B is the preferred drug for the treatment of invasive and life-threatening mycosis. What are the properties of polyenes? As you know, the polyene structure constitute a large macrolide ring of carbon atoms closed by the formation of an internal ester of lactone. This you will be seeing in the next figure. Then the macrolide ring structure, it constitutes about 12 to 37 carbon atoms with alternating conjugated double bond entirely in the cyclic lactone. This structure also contains a number of hydroxyl group on alternate carbon atoms, and these are distributed along the macrolide ring. The presence of free carboxyl group and a primary amine group on amphotericin B gave the compound its amphoteric properties as reflected by the name of this compound. Polyenes are soluble in polar organic solvent such as dimethyl sulfoxide. It is known as DMSO, but polyenes are slightly soluble in water and non-polar solvents. As you can see the structure of amphotericin B, you can see the NH2 and also you can see nystatin. You could see NH2 amine group, which is the reason which gives the amphotericin B and nystatin its amphoteric properties. You could see a large and long aliphatic chain in both of these structures. Biotherapy of fungal diseases. In this one, we will first discuss amphotericin B, whose structure you had just seen in the last slide. Amphotericin B formulations are extensively used for curing disseminated candidiasis, invasive aspergillosis, and cryptococcal meningitis. However, there are several issues with the usage of amphotericin B because these issues include such as solubility in water and toxicity limits, which limits the effectiveness of amphotericin B for therapeutic purposes. There are certain novel drug formulation of amphotericin B which are coming up. These are lipid formulation such as amphotericin B lipid complex, amphotericin B cholesterol sulfated, and liposomal amphotericin B. These lipid formulation, especially the lipid formulations of amphotericin B, differ in their pharmacokinetics, tissue distribution, and toxicity profile. Now, coming to the mechanism of polyenes. Polyenes group of compounds increases the permeability of cell membranes, and that will lead to leakage of cellular constituents and ultimately cell lysis and cell death. These polyenes at their fungicidal concentration can cause extensive oxidative damage to fungal plasmalemma. These antifungals bind very selectively to the membrane sterol, ergosterol in fungal cells and cholesterol in the mammalian cells. So this binding actually ultimately leads to the change in the cellular constitution of the cell, and further it will lead to the cell death. Antifungal agent amphotericin B causes formation of pores in the fungal membrane, leading to the leakage of vital cytoplasmic components and cell death. The activity of polyenes. Polyenes are highly effective, especially against Candida species, Cryptococcus neoformans, and the combination of amphotericin B and flucytosine, which is the next class of antifungal agents, a combination of these two, amphotericin B and flucytosine, or known as fluorocytosine, has additive or synergistic effects against different target cell types. Coming to the next class of antifungal agents, azoles constitute the second class of antifungal agents, and it is one of the most rapidly increasing groups of the antifungal agents. The first azole, which was discovered in 1944, was benzimidazole, and it was having a broad range of activity against several yeast and as well as the bacterial species. And till now, about 100 compounds of this class has been known to be synthesized, and many more are still in the developmental stage. And the prominent ones which are in this family is miconazole, iconazole, and ketoconazole. So these are the well-known antifungal agents. And next is clotrimazole and miconazole are helpful in combating significant human fungal infections. Phenylethyl-imidazole derivatives are known to be very much persuasive against fungi, dermatophytes, and gram-positive bacteria. Ketoconazole has led to successful treatment of oral candidiasis, coccidioidomycosis, endemic mycosis, dermatophyte infections, and veterinary fungal infections. Subsequently, many antifungal agents have been developed in this class of azoles. The structure of the azole family of compounds. These antifungal agents have 5-membered organic rings that include either 2 or 3 nitrogen molecules, especially in the imidazole and triazole respectively. The ring structure in both imidazole and triazole hold a short aliphatic chain in which the second carbon is linked to the halogenated phenyl group. Imidazole are soluble in organic solvents. However, clotrimazole and itraconazole are insoluble in water. Several family compounds of azoles are being listed here. You can see miconazole, ketoconazole, triazole family, voriconazole, fluconazole, and revocanazoles. So if you can see, this is a large aliphatic chain with phenyl group. Azole. Azole is antifungal agents. They mediate their antifungal activity by inhibiting cytochrome P450 enzymes. These are very well known cytochrome P450 metabolic enzymes, and especially the enzymes involved in catalyzing biosynthesis of ergosterol are targeted by azoles. What they do? They inhibit cytochrome C oxidative and peroxidative enzymes and affect the lipids of these cell membranes, which eventually leads to the leakage of proteins and amino acids from the cell. Azoles also inhibit enzyme catalase, a very well-known enzyme in the antioxidative pathway, catalase, and it decreases fungal adherence significantly by inhibiting germ tube and mycelium formation in Candida albicans. Fluconazole and Echinocandins can also affect the reduction of obtusifolone to obtusifilone. This very step occurs in ergosterol biosynthetic pathways, and inhibition of this pathway will eventually result in accumulation of methylated sterol precursors. Ketanazole inhibits energy generating ATPase pathway. I think you are familiar with the ATPase pathway. These are highly adenosine triphosphate generating ATP, generating energy pathway, especially in Candida albicans. These are targeted, and other yeasts, these are targeted by the ketoconazole. And what happens when the energy pathway is targeted? So it will lead to disintegration of the electrochemical gradient and reduction in the ATP levels. Several other enzymes such as chitin synthase, adenyl cyclase, and 5-nucleotidase, they are also inhibited by miconazole and ketanazole in fungal pathogen Candida albicans. These azoles at their fungistatic concentration can cause extensive changes in the plasma membrane and holes in the nuclear membrane. The cell membrane changes, they are especially more severe at the fungicidal concentration, and it includes disappearance of mitochondrial structure, which is eventually, mitochondria, this is the seat of highly energy generating pathways in the cell, and once they are targeted, you could assume it will lead to complete loss of energy generation and nuclear pathway. So coming to the treatment by these antifungal agents, especially for dermatophytes. So if you can see, dermatophytes can be treated either topically or systemically, and commonly used topical agents, as you know, is imidazole. These are being used for the treatment of dermatophytes. And how they treat the dermatophytes, used in the treatment of dermatophytes, basically they inhibit C14 alpha dimethylase, that is a cytochrome P450 enzyme, thereby blocking the demethylation of lanosterol to ergosterol. So this will disrupt the membrane structure and function, and thereby very much it will significantly inhibit the fungal cell growth. As you know, there are several limitations in the usage of azoles because several side effects have been reported, especially in hepatic, endocrinologic, hematologic, and gastrointestinal toxicity have been found to be involved with the usage of azoles, especially nausea, vomiting, that can occur when you use carbamazole for fungal infected patients at their conventional doses. And these are the very well-known side effects of ketoconazole. However, there are other side effects also, and these include headache, abdominal pain, diarrhea, non-fatal urticaria, exfoliative dermatitis, fever, fatigue, and anaphylaxis. So these are the several side effects of azoles. So these are the limitations which make them not so effective antifungal agents. And one important thing, these toxicity may be associated in a very much dose and time dependent manner. Another class of compounds which are antifungal agents, and these are pyrimidine analogues. Flucytosine, or known as fluorocytosine, is a well-known pyrimidine analog and a very well-known antifungal agent. It is a synthetic fluorinated pyrimidine. It was discovered in 1964, and this discovery was made in the very well-known famous Roche labs. And these Roche labs have their drug discovery program, and in the drug discovery program they were isolating and synthesizing the compounds which could be very much effective as cytostatic and anti-neoplastic agent. And this flucytosine or fluorocytosine was first discovered as a cytostatic and anti-neoplastic agent. But however, when they tested it, they didn't find these activities. But one important thing which they find was that though it may lag these cytostatic and anti-neoplastic activity, but it possesses antifungal activity, especially against Candida species and Cryptococcus species, and that led to its development, and right now it's very widely used against these species and fungal agents like Candida species and Cryptococcus species. And also they are also used in a limited number of modes such as Aspergillus species and agents of chromoblastomycosis. The chemical formula of flucytosine is 4-amino-5-fluoropyrimidine. It has molecular weight of 129 kilodalton. This compound is odorless, white and crystalline. Mechanism of action of flucytosine or fluorocytosine. This flucytosine or fluorocytosine exhibit its antifungal activity by two mechanisms. One is disruption of RNA and DNA synthesis, and the second one is alteration of amino acid pool. How it disrupts the DNA or RNA synthesis. This is the flucytosine is transported into the fungal cell by cytosine permeases, where it gets deaminated to 5-fluorouracil by enzyme cytosine deaminases. Now, since it got deaminated, it gets converted to 5-fluorouracil, which further is converted to 5-fluorouridylic acid by enzyme uridine monophosphate pyrophosphorylase. As the name suggests, pyrophosphorylase. So this enzyme is phosphorylate fluorouracil to fluorouridylic acid monophosphate. Then further phosphorylations are carried out, and these phosphorylated compound is then incorporated into RNA, which lead to the disruption of protein synthesis. That the humans do not possess cytosine deaminase, and replacement of uracil by 5-flucytosine in fungal RNA brought changes in amino acid pool of the fungal cells. The conversion of 5-fluorouracil to 5-fluorodeoxyuridine monophosphate is another significant conversion that will inhibit the thymidine synthesis, because this particular enzyme is involved in DNA synthesis and nuclear division. So you can see in this slide particularly. So we'll begin with 5-fluorocytosine, or denoted as 5-FC. So first is the deaminase, the action of the enzyme deaminase. So what does it do? It converts 5-fluorocytosine to 5-fluorouracil. So now the 5-fluorouracil is formed. It will be phosphorylated. So if you can see in the next step, particularly pyrophosphorylase, it is phosphorylated by enzyme nucleoside phosphorylases, which leads to the formation of 5-fluorouridine monophosphate, or denoted as 5-FUMP. Now this phosphorylated compound again undergoes the action by enzyme kinases. You know the enzyme kinases, they are very well associated with the phosphorylation. So again this compound is phosphorylated. So this phosphorylated compound is known as 5-fluorouridine triphosphate, as the name indicates, triphosphate. So there are then 3 phosphate groups in it, if you can see in the structure, right? So then this phosphorylated structure is incorporated into the by RNA polymerase, and it will lead to the disruption of RNA synthesis, and its inhibition in the DNA synthesis has also been discussed in the previous slide. So that is how 5-fluorocytosine inhibits the fungal infection, and by converting these metabolic pathways, by using these metabolic pathways, by converting 5-fluorocytosine to 5-fluorouracil. So now coming to the activity of 5-fluorocytosine, it exhibits a broad activity against Candida, Turulopsis, Cryptococcus and Trichum genera. It is also, though effectively in a moderate manner, against Aspergillus and Chylomycosis, causing fungi. It has shown both fungicidal as well as fungistatic activity against several fungal pathogens such as Candida albicans, Candida tropicals and Candida neoform. However, against side effects of 5-cytosine, it causes gastrointestinal effects such as diarrhea, nausea, vomiting, and bone marrow suppression also develop following 5-fluorocytosine treatment. 2-cytosine, standard of the care for the treatment of cryptococcal meningitis. The combination of 5-fluorocytosine with amphotericin B is recommended for treating central nervous system candidiasis and cryptococcus. Then coming to the fourth group of antifungal agents, these are allylamines and thiocarbamates. Terbinafine is a synthetic compound and only allylamine in clinical use. It is derived from topical antifungal naftifine, which is considered to be the original compound of allylamine class. Tolnafetate is the only thiocarbamate available. Terbinafine and naftifine have the chemical structure similar to a naphthalene ring substituted with an aliphatic chain. You will see in the next slide. You can see the class of these compounds, naftifine and terbinafine. You look at your naphthalene ring substituted with an aliphatic ring, and then subsequently you can see another class of these compounds. So these have long aliphatic chains are present in these family of compounds. Terbinafine, it acts as a non-competitive inhibitor of enzyme squalene epoxidase. Non-competitive inhibitor, it acts as an inhibitor in comparison to the substrate, but in a non-competitive manner. This enzyme, squalene epoxidase, catalyzes the formation of lanosterol from squalene. It is considered an essential pathway in the biosynthesis of fungal cell membrane. So once this pathway is targeted, what will happen? It will lead to an increase in squalene concentrations due to disruption, which will increase the cell permeability as well as disruption of the cellular organization. And finally, or eventually, it will lead to the cell death. Terbinafine is a well-known antifungal agent against several dermatophyte infections, including onychomycosis. Naftifine hydrochloride is effective against Tinea cruris and Tinea corporis. Terbinafine shows limited success against invasive fungal diseases, but when we use combinatorial therapy, in combinatorial therapy, especially with voriconazole, it will be very much successful and effective in treatment of infections caused by Pseudosporium prolificans. Now coming to the next group of compound, Echinocandins. So this is a fifth class of antifungal agents, and as you know, fungal cell wall contains several compounds that includes menin, chitin and glucans. Now, a series of compounds have been developed in the drug discovery program of several companies that can effectively target these cell wall compounds. So especially three group of compounds, namely leucines, echinocandins and papillocandins, act as inhibitors of fungal 1,3-glucan synthesis. This is the enzyme they are going to inhibit. So echinocandins consist of semi-synthetic cyclic lipohexapeptides, and they have a molecular weight of about 1200 daltons. Thiopungin was the first echinocandin used in the clinical trials, but its development was abandoned because of the toxicity. In the late 1990s, three echinocandin compounds, namely caspofungin, micafungin and anidulafungin, has been found to be active against Candida species and Aspergillus species, and these compounds, which are undergoing development, have undergone clinical development and they have been evaluated further. So you can see these group of compounds, these three group of compounds. These are capsulofungin and micafungin and anidulafungin. So you can see the big structure of these compounds. Mechanism of action of these echinocandins. As you know, these echinocandins act as non-competitive inhibitors of cell wall protein enzyme 1,3-beta-D-glucan synthesis. And since they can act as non-competitive inhibitors, it means they are not in competition with the substrate. So they act as a non-competitive inhibitor, right? And because of this very inhibitory action, they're going to exert secondary effects on other components of intact cells, such as reduction in ergosterol and lanosterol content. Decreased 1,3-beta-D-glucan concentration in the cell wall will result in osmotic fragility and lysis of Candida species. Although Echinocandins do not kill Aspergillus species. They bring on morphological changes within these fungi and reduces their tendency to cause pulmonary injury. So, students, let's summarize from what we have learned in this module. As you know, in spite of several advancements in the recent past as well as in the coming future, especially in the antifungal therapy, there is still demand for new antifungal agents, as well as there's a need for better treatment strategies with them. So, it is very much desired that this new antifungal agent does not have a broad spectrum of activity, but tremendous activity, especially against the two well-known fungi, Candida and Aspergillus, because these fungal agents still has a high mortality rate. Diseases caused by these fungal agents has high mortality rate. Triazole and echinocandin resistance is of concern, and therefore more rapid approaches to detect and continuous surveillance are required, especially in the antifungal therapy. The combinatorial therapy has also to be looked after. This combinatorial therapy of several antifungal needs to be revisited to maximize the survival rate and minimize the tissue damage caused by fungal infections. Thank you.